students, today we will learn about the starter cultures in food and dairy products. Under this, we will learn about the different type of starter cultures, pure, mixed starter cultures, single or multiple strain starter cultures or mixed strain starter cultures. We will also learn about the back sloping and classification of the starter based on their metabolism. Also, we will learn about a very important group of microorganisms. In this, we will also learn about the lactic acid bacterial starter cultures, the mold starter cultures, the general principles of culture maintenance and their preparation, the criteria for the selection of cultures to be used as a starter, and how the starters are prepared and how the starter cultures are utilized for producing various fermented products such as sausages, cheese, etc. To begin with, we should know what is a starter culture. A starter culture is an active microbial preparation deliberately added to initiate desirable changes during the preparation of a fermented product. The starter consists of a cultivation medium that have been well colonized by the microorganisms which are an integral part of the fermentation process. Microorganisms used in the starter include bacteria of different types such as Enterococcus, Lactobacillus, Lactococcus, Streptococcus, Leuconostoc, Pediococcus, Lactobacillus species, Acetobacter, and different fungi such as Rhizopus, Aspergillus, Mucor, Saccharomyces, and different yeast. The practice of retaining a small quantity of whey, cream or sourdough from a successful fermented product and its usage as an inoculum or a starter for the next batch of production is termed as back sloping. Let us learn about the different type of starter culture in food industry. First, single strain starters. As the name indicate, it consists of only one pure microbial culture. Single strain of Streptococcus cremoris or Streptococcus lactis is used either in pairs or as single inoculum. The second category is multi-strain starters. These are well-defined mixtures of 3 or more distinct strains of organisms. Examples include Streptococcus cremoris, Streptococcus lactis, Leuconostoc and Streptococcus diacetyl lactis strains. The third group of starters are mixed strain starters. It consists of a mixture of strains of Streptococcus cremoris, Streptococcus lactis, Streptococcus diacetyl lactis and Leuconostoc. The identity of the constituent strain is frequently unknown to the user and their composition may vary over subculturing process. And a new terminology for the classification of starters has been coined. First, defined starter cultures, which include identified strains and species of the component strain. Second, undefined. These include artisanal cultures which are traditionally used. Depending on the metabolic activities, the starter culture includes mesophilic starter cultures when the optimum growth temperature of the organism is 30 degrees C, the thermophilic starter cultures where the optimum growth temperature of the organism is 45 or more. Depending on the physical state, the starter cultures can be liquid, solid or powder. Prior to the selection of organism to be used as a component of the starter culture, it should satisfy certain criteria. The important criteria for the selection of microorganism as a constituent of starter culture is: first, they should produce flavor and they should not produce off flavors in the final product. It should exhibit salt and bacteriophage tolerance. The starter culture may produce exopolysaccharide if it is required in the final product. The starter culture should produce bacteriocin and should exhibit temperature sensitivity. The starter culture used should also bestow functional health properties, and it should enrich the food with respect to vitamins, probiotics, prebiotic, oligosaccharides, bioactive peptides and lipids, mannitol and folate, etc. These days, there is another group of starter which are known as starter concentrates. These are direct vat set, also abbreviated as DVS, or direct vat inoculation, DVI. These cultures replace the bulk starters. DVI culture are concentrated cell preparations containing a very high count of 10 raised to power 11 to 10 raised to power 13 colony forming units per gram of the starter. These are available in two physical forms, as frozen pellets or as in freeze dried granular forms. The major advantage of the use of DVI is reduced probability of contamination from undesirable organisms. Secondly, no requirement of incubation, or there is no need of subculturing. However, there are some major disadvantages, that is restriction of the number of strains suitable for the concentrate production, requirement of extremely low temperature storage facilities, and the high cost involved in the production of DVS cultures. Lactic acid bacteria are commonly used as starter culture in many food fermentation processes, and they are common in dairy fermentation. Some members of lactic acid bacteria like Enterococcus, Lactobacillus, Lactococcus, Leuconostoc and Streptococcus are the commonly used lactic acid bacteria as starter culture. Selected examples of starter cultures are Brevibacterium linens. It is utilized for flavor production in Limburger cheese, Leuconostoc mesenteroids, and lactic producing flavor for the conversion of glucose, fructose to CO2. For example, in Emmental cheese, Lactobacillus delbrueckii in conjunction with Streptococcus thermophilus for its use for yogurt production. Thermophilic cocci are also used as starter for yogurt, mozzarella and cheddar cheese. Bifidobacterium longum, Lactobacillus acidophilus, Lactobacillus delbrueckii and Streptococcus thermophilus synthesize folate and secrete into the medium during the fermentation process. Some mold cultures, that is some fungi, are also utilized as starter culture in various food fermentation process. The most important example are the Penicillium species, which are utilized for fermentation of meat products. Penicillium camemberti is used for white cheeses. Penicillium roqueforti, it is utilized for the production of blue vein cheeses. There is another type of starter culture which has been utilized since ancient time. That practice is known as back sloping. In this process, high initial counts of the desired microorganism, whether it is pure or mixed culture, are utilized. These are active in the fermentation without compromising the quality of the product. The organisms isolated from the surfaces of the smear ripened previous cheese, brine tanks and other sources of plant include Microbacterium, Brevibacterium and film forming yeast. The advantage of this process is that the antimicrobial substances released by the starter organism discourages the growth of undesired microorganism. The disadvantage of this process is its potential to magnify the load of undesirable microorganism to high level if the original source is contaminated. There is another term which is known as artisanal cultures. These are natural milk artisanal cultures which comprise of thermophilic lactic acid bacteria. These are low cost starters with undefined composition as well as performance. These are prone to undesirable contamination and are associated with microbiological hazards. Backsloped starter artisanals utilize some of the previous product batch as a starter. The advantage is that it is low cost and it is also of undefined composition. Therefore, they require careful handling and quality control testing. Whey cultures are used on industrial scale for making some traditional Italian and Swiss cheese type. This is an example of the artisanal starter cultures. Now, let us see what are the steps in the production of some of the fermented products using the starter cultures. So, the steps required for the cheese preparation are: first, acidification of the milk by the starter and the rennet; second, milk protein coagulation, followed by separation of curd from the whey, and then ripening of the cheese take place. Similarly, starter cultures are also used for bread making. There are two pre-ferment varieties: sponge, where the yeast Saccharomyces cerevisiae is used as a starter culture, and second, sourdough, where wild yeast, lactobacillus and acetobacteria present in the grain bring about the fermentation. During the bread making process, the starter are performing important functions. They convert the sugar to carbon dioxide to create a light and airy texture, improve the flavor by production of alcohol, and they also bring a change in the protein structure of the bread. Starter cultures are also utilized for the fermentation of meat to produce sausages. The prime starter culture organism are lactic acid bacteria, which bring about the fermentation, and the prominent organisms are lactobacilli, Lactobacillus plantarum, Lactobacillus pentosum, Lactobacillus curvaceus, Lactobacillus farciminis, Pediococcus, Pediococcus pentosius, Pediococcus acidilactis, etc. Curing bacteria for the color and flavor forming organisms are bacteria of Staphylococcus and Courcia species. There are certain yeast like Candida and molds like Penicillium which also help in the curing process. Let us see what are the different steps for the conversion of raw milk to cheese. The first step is pasteurization, wherein the milk is pasteurized. This step is followed by addition of a starter culture and rennet. The process results in the formation of curd and whey. The product so formed is cut, scalded, pitched and drained, resulting in the formation of curd. The curd is added with some salt and the process is known as seddering, milling and salting. This yields raw cheese. The raw cheese is pressed, molded, matured and packaged to form the mature cheese. Starter cultures are also utilized for the yogurt fermentation. The process involves pasteurization of the raw milk and then starter is added. The milk is homogenized, cooled and incubated. It results in raw yogurt. The raw yogurt can be utilized directly to form processed yogurt, or we can add fruits or flavors to fortify the raw yogurt to form different type of products. An example of mixed type of starter culture is jikwa. It is a complex mixture of molds such as Aspergillus oryzae and Rhizopus oryzae, yeast, for example Saccharomycopsis fibulifera, bacteria like Bacillus and Lactobacillus, and their metabolites, primarily amylolytic and proteolytic enzymes, which are cultured on a starch-rich substrate in a solid state fermentation. It is available as dried bricks, balls and powder form. This is a commonly used starter culture in China. Another traditional starter culture is koji. It is nothing but cooked rice alone or in combination with soybean beans, inoculated with a fungus Aspergillus oryzae. Koji is utilized as a starter in various fermentation products, for example miso, sake, vegetable pickles and soya sauce. So, students, let us sum up. Starter cultures are an active microbial preparation which are deliberately added to initiate a desirable change during the preparation of fermented products. They have multifunctional role in the food fermentations. Starter cultures are classified as defined or undefined. On the basis of metabolism, they may be grouped as mesophilic or thermophilic. The use of starter culture cell concentrations having high count of microorganisms are known as DVS or DVI, and these have replaced the bulk starters. During fermentation of raw material, the production of organic acids like lactic acid, acetic acid, production of alcohol like ethanol and aroma compound, bacteriocins, exopolysaccharides and several enzymes contribute to the enhanced shelf life and microbial safety of the final product. They also improve the texture, sensory property of the end product. Lactic acid bacteria, which are commonly used as a starter culture, they also produce volatile substances which impart typical flavor to the certain fermented foods such as sourdough, kefir, komes, butter and buttermilk, etc. Bifidobacteria produce exopolysaccharide which acts as a viscosifying, stabilizing and emulsifying agent, traditional starter cultures like ku and koji have been used as fermentation starter for a large number of popular food even today. Thank you.